Okay, welcome to another week. So today we're going to pick up where we left off with our capillaries. So last time we finished up with arterioles, we had been talking about intrinsic and extrinsic factors that change the radius of the arterioles, therefore changing their resistance to flow. And we had talked a little bit about how that also can affect mean arterial pressure, which is going to be kind of like a measure of our blood pressure. And then we had moved into our capillary system. So we talked about how capillaries have thin little walls, and this is where we're going to have exchange with tissue. All right, so we were talking about a little bit about that anatomy of the capillary beds before we get into our main topic for today, which is something called net filtration pressure. Okay, so we want to know that our capillaries are thin. They just have one cell layer. So it's a layer of endothelial cells. There's also a basement membrane like sticking them together, but just one cell layer, which means they have a really small barrier for diffusion. So things can get across really easily, which is great because that's the job of the capillaries, to allow for exchange. Okay. All right, so we know that we have lots and lots of capillaries, and their surface area is crazy big, which is good. Again, allowing for them to have a lot of room for this exchange with the tissue, and they have pores between them. Okay. So when we left off, we were looking at some of the ways we can control flow through the capillaries. So we have our arterioles going into one end of our capillary bed. We're going to have a venule coming out the other side. And we were thinking about these other two types of structures called our meta-arterioles, which is what we talked about when we left off. We're going to take a quick look at the precapillary sphincters. Okay. So these two concepts are relatively simple, hopefully. It's just that sometimes we're going to want blood to flow through capillaries in a specific region, and sometimes we're not going to care that much. We're going to want to bypass those capillary beds. So a meta-arteriole, when it's nice and open, allows you to kind of bypass and rush blood past that capillary bed. So in order to be open, right, we would want it to have a relatively large radius, therefore low resistance, therefore plenty of flow. You can see that it's bigger than the capillaries, so blood would tend to prefer to go through that meta-arteriole, at least some of it. You functionally still have some flow through the capillaries, but less than you would if it were narrowed and closed. So if you narrow the radius of that meta-arteriole, you pump up its resistance, it's going to force blood into the capillary networks around it. Okay. So functionally, this works because there are rings of smooth muscle around the meta-arteriole at strategic locations that allow you to kind of block it off or open it up, depending. So if the meta-arteriole contracts, blood is not going to go through the meta-arteriole, it goes through the capillary instead. So contraction of the smooth muscle in a meta-arteriole formally increases blood flow through the capillaries. And the reverse is true. If that smooth muscle relaxes, that means the meta-arteriole is dilating, it's getting a larger radius, therefore lower resistance, more flow, which means that if the flow is going through the meta-arteriole, relatively, you'll have less going through the capillary. So a somewhat related concept or a similar concept. So we also have a structure called a precapillary sphincter. So this is again going to be a ring of smooth muscle. So we can see those on here. So here we see the little swirly little curly Q strand that's supposed to be smooth muscle surrounding this beginning of this capillary. So it's a ring of smooth muscle. It's on the arteriolar end, right? So where blood would initially be coming into the capillary. So if that contracts, shuts that door, again, we're going to cut off access to that capillary bed. So that would stop flow through that capillary bed. If it relaxes, opening up that door, opening up that passageway, that would increase flow through the capillary bed. Now, these are only responsive to local factors. So we talked about types of things we mean when we say local factors. So that would be things like that reaction to oxygen and carbon dioxide concentrations in the surrounding tissue that we talked about when we were comparing active hyperemia and reactive hyperemia. So that kind of thing. Yeah. So here we can see that our metabolites, so like excess carbon dioxide would be a metabolite. So our waste products from respiration would cause relaxation of that smooth muscle of that precapillary sphincter. And that's because we'd want to get rid of excess carbon dioxide. We'd want to get rid of metabolites. So we're trying to have more blood flow through that area. So fundamentally, this is just kind of controlling when do we want a lot of exchange to happen in this region and when do we not need much exchange. So it's similar to what we talked about when we talked about our arterioles. Okay. So our big important topic for today, and our big important topic, honestly, for capillaries is filtration and exchange. So we'll get into what I mean by all of that, but we're going to have a couple different ways that things can cross capillary walls. And the one we're really going to be focusing on most is diffusion. So diffusion is just basically a passive movement, right? So things that are lipophilic, so they like lipids, so non-polar things that can get across membranes. So we talked about the properties of membranes, the properties of phospholipids, the properties of that phospholipid bilayer in A&P1. So anything that could get across our cell membrane, right? So we're thinking largely our non-polar gases is usually what we're talking about there. So things like oxygen, carbon dioxide, they're going to be able to go across the membrane. Things that are lipophobic are going to need a channel to help them out. So we're going to need an embedded protein somewhere, creating a hole in our membrane in order to allow things to go across. But diffusion is a passive process. When we talk about something called exchangeable proteins, proteins are larger. Often, thus far, we've been talking about moving things that are small. Proteins are big. So you want to think about the exchangeable proteins, kind of like the class of like our neurotransmitters. That was another big thing that we moved. So they're going to have to move via transcytosis. So we're going to have to like actively surround them and allow them to move. We talked a little bit, honestly, not much, because we didn't talk that much about our glial cells in our neuro unit, but there is also mediated transport in capillaries across the thing, in the brain. Okay. So mediated meaning kind of like controlled. Okay. But the main thing we're thinking about for our purposes for this unit, and a big concept to understand as we go forward, because this will kind of come back, especially when we get into the renal unit and think about kidneys, is the fact that we're going to have fluid flow depending on pressure gradients. Okay, so we've talked about fluid flowing through the cardiovascular system, through the vasculature or blood vessel, based on a pressure gradient. Now the pressure gradients we're thinking about are going to be across the capillary wall. So we're now thinking about when does fluid go into your tissue? So we might be thinking about eventually things like swelling, right? That would be important to understand this filtration process first. When we get to the kidneys, we're going to talk about their role in actually filtering your blood, sort of adjusting concentrations of things, getting rid of waste. Okay. So this is one of the important roles of capillaries throughout your body. And especially when we get to our kidney unit, we'll think about a modified version of this a lot. Okay. So the fluid we're talking about, it's basically plasma. So we know that plasma is one of our types of extracellular fluid, right? So we had our plasma and we had our interstitial fluid. Right? So we're thinking about when does that fluid, that plasma, move across a capillary wall. Usually proteins are not moving with the plasma. The proteins are usually too big. They can't get across the capillary wall. And they're actually going to exert a certain type of pressure on the fluid. Okay. So we're first going to talk about directions before we get into those little pieces. Okay. So if we have a capillary, I'm just going to draw this as our blood vessel. I can even draw a circle at either end. I'm going to make one end blue because I'm going to make the red end our arterial end, the blue end, the end going towards the veins. Okay. We're also going to draw some tissue. Just over here, this is going to be tissue. So we got some cells of some type over in here. Okay. So what we're talking about, tissue. This is our capillary. Okay. So we have some plasma in our capillary. That's our fluid here. And we have interstitial fluid by the tissue. So this is technically our interstitial, so between tissues, interstitial, between tissues, interstitial. So this is where our interstitial fluid would be. So CF in that interstitial space. So the two types of movements we're talking about is when we move fluid from the plasma into that ICF, so into that interstitial space. This is filtration. So that arrow I just drew, filtration. And the reverse is absorption. So if we have fluid in this interstitial space, we have movement from that area back into the capillary. That's going to be absorption. So fundamentally, what we're going to be thinking about as we're thinking about exchange of fluid across capillaries, so we're going to be thinking about, are we going to have overall, so on net, that's where the net from net filtration pressure comes from. Overall, are we going to have more filtration, so movement towards that interstitial space? Or are we going to have more absorption, so more pressure moving fluid back into the bloodstream, back towards the capillary? So we're going to be thinking about factors that influence these two directions because they're kind of in competition, right? The fluid can actually only move overall in one direction, but we're going to have forces pushing and pulling on it. Okay. So our overall purpose here is to appropriately distribute our extracellular fluid. Our extracellular fluid has the two parts that we did on like the first day of A&P 1, I think, the different parts of water in your body. So we had the plasma versus the interstitial fluid. Oops, I drew. Sorry about that, guys. Interstitial fluid. Sometimes I can't spell. Anyway, so those are two types of fluids. Okay. That's probably why we wrote it onwards. Okay. So we're trying to appropriately make sure that we have the right amount of fluid around our tissue, the right amount of fluid in our bloodstream. So we will eventually have an equation, but I'm just going to let you know ahead of time as we talk about these factors that what we're ultimately solving for, our net filtration pressure, is just going to be, okay, how much force do we have for filtration? Right? So how much force is there for moving towards that interstitial space versus how much force is there for that fluid to come back into the capillary? So we're going to subtract out our absorption. All right, so this is Either fluid moves out or fluid moves in. So we're just going to add up forces for it moving out and subtract out the forces for it moving in. So conceptually, that's what we're doing as we move through the next bit. Okay. So here we have a picture of our capillary, and this is just some of the movement of things that would have gone across. So when we're talking about like lipid soluble substances, so lipophilic things, I mentioned our gases, the carbon dioxide and the oxygen, as things that can move across those walls through diffusion. And those will be especially important when we talk about the lungs as well, which will have a similar cell layer because in your lungs you have blood and air nearly in contact. But there are other things, fatty acids, steroid hormones, are exchangeable proteins. Here you can see the transcytosis. So first endocytosis, pulling it into a little membrane bag, then exocytosis on the other side. And we can see some things going through pores between our cells. All right, so you can see it's going between the cells along with the water. So it's going to be water soluble substances. So some of our ions that we're thinking about, potentially glucose, could go through a pore depending on where we're talking about. Okay. All right. So back to this idea of filtration and absorption. Okay. So we're going to have two types of forces. It says just two forces, but we're actually overall going to be adding and subtracting kind of four things. So we're going to say that we have two types of forces for now. We're going to have hydrostatic pressures, and we're going to have osmotic pressures. Okay. So hydrostatic, hydro meaning water, static, I think that means like place or something. I'm not exactly sure, but this is going to be pressure coming from the fluid, like the push. So we've been looking at lots of fake little water columns as we've been talking about pressure gradients across a blood vessel. This is basically what we're talking about when we're talking about hydrostatic pressure as well. So what we're talking about is just if we have a tube, instead of doing a column, we're going to have a tube because we're going to draw our capillaries as tubes. The most important one we'll eventually be talking about is just the fact that blood pushes on the walls of the capillary as it moves through. So that would be an example of hydrostatic pressure. It's just the force of the fluid on that wall. Okay. So given the directions I drew those arrows, someone want to tell me, predict whether hydrostatic pressure of that blood in the capillary is a force for filtration or absorption? What direction was filtration? Out. All right. So what direction are those arrows going? Out. Yeah. So this is going to be one of our main forces for filtration on a future slide. Right. So anything that pushes us in that direction is going to be a force for filtration. Any arrows that we draw in are going to be our forces for absorption. Okay. All right. So that's what a hydrostatic pressure is. Okay. So an osmotic pressure, remember osmosis. Anybody want to tell me anything that they remember about osmosis? Yeah, movement of water, right? So we were looking before at cells, and if we wanted to think about the movement of water across a cell, what other information might we want for that? To figure out what direction water would move across a cell, what else would you put in here? Yeah, so when we're thinking about osmosis, we're thinking about molecules or solutes inside a cell versus outside a cell, and essentially their relative concentration gradients, right? So if I'm drawing whatever they are, let's pretend for our purposes right now, because we're about to talk about capillaries, let's think about these as actually proteins, right? So we have water moving, partially dependent on the concentration of other molecules inside the cell versus outside the cell. So in this picture I just drew, I want you to predict in your mind, which way would the water go? So is water going to go towards more solutes or towards less solutes? So water is actually, when we're thinking about osmosis, we're trying to get like an even spread of solutes. We're trying to reach equilibrium, right? It's just like kind of how the world works. We want, your cells quote unquote want just naturally to have equal distribution, equal concentrations on both sides. So if we're thinking about concentrations of solutes, water moves in whichever direction is going to leave them evenly spread out and at even concentrations. So water usually, in this example, is going to move towards the greater concentration in order to push them farther apart. Although I did draw them kind of evenly spaced, I drew more. So maybe let's pack them together, right? Because we want them to spread out so everything's evenly spaced. Yeah. So that was our sort of basic idea of osmosis before. And that's where our osmotic pressure is coming from ultimately. So our osmotic pressure is going to be kind of the pull of these solutes on the water, right? So these solutes that we've drawn here, right? There are lots of them. They're pulling water towards them because the water wants to spread them out, water them down until they have the same concentration as whatever's inside that cell, okay? So we're thinking about hydrostatic pressure as the force of a fluid pushing out on the walls of its container. And we're thinking about osmotic pressures as the fact that there is stuff in a certain region and it's going to pull water towards it to kind of water down those solutes. So those are the two types of forces that we're going to be thinking about as we talk about our net filtration pressure. Okay. So specifically when we're talking about osmotic pressure, these need to be non-permeating solutes. So they need to be something that can't just cross the wall on its own, can't just cross through our membrane. Because if we have a solute that can cross the membrane, has no problem getting through that phospholipid bilayer, then it would just move on its own. It doesn't need the water to move. So it needs to be something that is non-permeating, that can't move across that layer of endothelial cells easily, because that's why it's affecting the water instead of its own movement. Okay. So the non-permeating solutes that we're thinking about here are proteins. They're too big. The only way we had to move them was through that transcytosis process, which is involved. We have to do the endocytosis, the exocytosis. That's a type of active transport requiring energy. So that's not going to happen just automatically. But this movement of water can happen just passively through osmosis. So that's the effect essentially of proteins in the blood and proteins in the surrounding tissue. Okay. We do use another word specifically when we are talking about osmotic pressure that is coming from proteins specifically. So there's just a tiny, tiny difference. Oncotic pressure is the osmotic force of proteins. So we can use these somewhat interchangeably for a little while because we're talking about the osmotic pressure of proteins in these specific places. But that's why there's two different terms that you'll see. Okay. All right. So when we're thinking about the hydrostatic pressure first, so I'm going to draw some capillary and some tissue again. Okay. So capillary, we have an arterial end, we have a venous end. That bend that I drew was just because my hand moved, not because it's important. And we got a bank of tissue. And we have this interstitial space here, this space between tissues. Okay. So for our hydrostatic pressures, they're going to get a big P as their symbol. And we're going to specify the pressure from the capillary or the pressure from the interstitial fluid. I'll just write IF here, because that's how your book indicates it as well. So hydrostatic pressure is going to be the big P as we build towards our equation. Okay. So for our capillary hydrostatic pressures, at the arterial end, where we have the blood coming in, we're going to have a pressure of 38 millimeters of mercury. But by the time we reach the venous end, we're going to be at only 16. So when we talk about our net filtration pressures, we are going to see some variation depending on where we're calculating our pressure, right? So sometimes we may be asking or thinking about, okay, what is the filtration pressure? Actually, maybe since we're talking about pressures, I'm not going to draw us an arrow. So the filtration pressure here versus the filtration pressure over here on the opposite end can differ, okay? So where we are can matter. Oops. Where we are can matter. Okay. So both of these, though, are positive numbers, right? So we have positive numbers for hydrostatic pressures. They are different amounts, but they're both pushing out because we're still just talking about blood pushing out on the walls here or here. Okay, so both of those are just capillary pressures. The only difference is if we've specified what end we're talking about. Okay. So here, they're both favoring filtration, each of them. So 38 on the arterial end, 16 on the venous end, but both of them are going to tend to push fluid towards that interstitial space. That's why we say they favor filtration, because they're pushing us in that filtration direction. Now, there is interstitial fluid. Not a lot of it. It's not high pressure, but it does exist. So we do have another hydrostatic pressure we can think about. So these were our pressure in the capillary. But we do also have pressure in this space between the tissue and the capillary. So in that interstitial fluid, okay, we have a tiny bit of pressure pushing us back, okay? So we have a tiny bit of fluid that's trying to push into the capillary, but under normal conditions, this is really low. So 0 to maybe 1 millimeter. Oops, that's just right, 0 to 1 millimeters of mercury. So under normal conditions, this is not very significant, but we do want to know it exists because we can potentially have things that change this pressure. We won't talk about many of them, but in disease states, it could happen. So we want to conceptually understand that it exists. Now, when we're thinking about the interstitial fluid, this is just going to be everywhere around the tissue. So it's not going to be different at the arterial end versus the venous end. It's just what our interstitial hydrostatic pressure will be. Okay. So we can calculate for each of these ends now a pressure gradient for our hydrostatic pressures. So we would just do that through subtraction, right? So we're figuring out how much force do we have for filtration? All right, filtration is the direction that's going towards this interstitial fluid, minus how much force do we have for absorption? That's anything pushing or pulling coming back towards the blood. So we just subtract 38 minus 1. We're going to consistently use 1 for the interstitial fluid so that we remember this pressure exists. So that's going to be our choice. So here our hydrostatic pressure gradient is going to be 37. So 38 minus 1. And here our hydrostatic pressure gradient on the venous end would be this hydrostatic pressure of the capillary at the venous end, 16 minus 1, giving us 15. But actually, questions at this point about the hydrostatic pressures? Okay. So we have pressure of blood.
The interstitial fluid that takes away from the capillary, like.
Yes, exactly. Yeah. So what we've done here conceptually, right, is forces for filtration. Which here was our pressure of the capillary. And we've subtracted forces for absorption. Which was our pressure of the interstitial fluid. And the reason I even draw it out for myself when I'm thinking about these things, because for me, it's easiest to just see that the arrows go in different directions and knowing that filtration is this direction, absorption is that direction. So that's why we have to subtract, because we're trying to find our net value. We're trying to find a total value. So yeah, exactly. This pressure, this hydrostatic pressure of the interstitial fluid takes away from the pressure, the hydrostatic pressure in the capillary. All right. So next, we're going to add on our osmotic pressures. Okay, so I'll kind of clear our slate. I'm going to redraw the pictures, but not the numbers, so that we can think about these separately. So again, capillary, we're going to have some tissue, and we have tissue up top. So direction words stay the same. So moving towards tissue is still filtration. Moving towards the capillary is still absorption. But now we're thinking about this pressure as a pull from dissolved proteins, essentially. So they're going to be pulling towards something. So really, if we draw the arrows, we can treat them just the same, but we're going to be thinking about what's pulling towards the tissue versus what's pulling towards the blood. Okay. So because these are both pressures, but we do like to write down little equations for ourselves eventually. I'm going to tell you that instead of a P, when we're talking about osmotic pressures, we're going to use a pi symbol. Okay. So the P is referring to the hydrostatic. The I is referring to the osmotic. So we're going to have osmotic or oncotic pressure of the capillary and of the interstitial fluid. Okay. So before we had P cap, PIF, now we have pi cap, PIF. Right. So each of these two spaces has a hydrostatic pressure and has an oncotic osmotic pressure. Okay. So when we're thinking about the capillary oncotic osmotic pressure, I'm going to be drawing solutes, so little dots for both of these. So anybody want to tell me where I am going to be drawing little dots right now when we're thinking about the capillary oncotic osmotic pressure? I'm about to draw for you. So we're saying that the capillary has a pressure, which is going to be a pull based on proteins. So where would those proteins be? They're going to be in the capillary, right? So what we're talking about, the capillary oncotic osmotic pressure, what we're saying is that we have stuff dissolved in the plasma, right? And this is producing our capillary oncotic osmotic pressure. And so what directional arrow would we draw here? We're going to be moving towards the capillary or away from the capillary based on those solutes. We're going to go in towards them, right? They're exerting a pull. Perfect. Okay. So we're pulling fluid towards those proteins. So that's why we say, and here they put it as reabsorption. We can talk about why sometimes we say absorption and reabsorption later. Fundamentally, we're still talking about the same thing for our purposes right now. So that direction back into our capillary is the absorption direction. So we had fluid pushing out, hydrostatic pressure out towards the interstitial fluid from the capillary, but we have the proteins in the plasma pulling fluid towards them. So we have those two things, those two capillary forces fighting against each other. We do have numbers to go with each of these. So our capillary oncotic osmotic pressure is about 25. So this force overall favors reabsorption because we're talking about going into the capillary, and that's what that direction is. Now the interstitial fluid also has some stuff dissolved in it. Again, this is something that can vary in disease states, but is usually quite low. Okay. So a little bit of stuff. Here, we're going to pick up the 0 for right now, but it would pull materials towards it in the same way that these proteins were pulling fluid towards them. Right? So there is a theoretical pull of those dissolved proteins in the interstitial fluid towards them. So anytime we draw an arrow from the blood towards the tissue, that's our filtration direction. Okay. So we would say that our interstitial fluid osmotic oncotic pressure, which is abbreviated pi IF, equal to, here we put 0, right? Essentially somewhere in between here, but it favors filtration when we have any number there, because this is the filtration direction moving out towards the tissue. The absorption or reabsorption direction is back towards the blood. So we can calculate a pressure gradient here as well for our osmotic pressure gradient. So we're going to, here we know that our force for reabsorption is greater. So they've calculated it as a force for reabsorption, right? Which is why they've done 25 minus 0, because you can just see on paper which is bigger and know. But you could do it the opposite way if you were trying to find a filtration pressure. You do 0 minus 25 and get a negative 25. Okay. So if you know first that you're going to favor reabsorption, you can just do the 25 minus 0, 25 for reabsorption. But when we are calculating and we don't know what the overall situation is, it's standard for us to calculate filtration pressures. So as we start to put these equations together, we saw that that hydrostatic force was for filtration. So if we just treat everything as if it's filtration, we want our filtration minus absorption our force for filtration is the direction out of the capillary. So that would be 0. And then we'd subtract our force for absorption, which would be 25. And we'd get a negative 25. The reason I like to do it this way, and I'm just showing you that additional thing that's not on the slide, is because I find it helpful to calculate it out the same way. And then I know if I ever get a negative sign, that's how I know absorption happened instead of filtration. So if you can know that without doing the math, great. But if you have to do the math, I like to think about that. Am I getting a positive? Am I getting a negative number? It's helpful for me. Okay. So our fluid is responding to both of these types of forces, both the hydrostatic pressures and the osmotic oncotic pressures. Okay, so we're going to now put them all together. We're going to have a capillary with an arterial end and a venous end, and we're going to start by calculating at the arteriolar end, because we saw that one of these forces varies depending on which end we are. Somebody want to remind me which of these forces varied from end to end of our capillary? Let's run through them. Did the hydrostatic pressure of the interstitial fluid differ? That pressure from fluid between the tissue and the capillary, did that change? No, not that one. What about the pressure, the hydrostatic pressure in the capillary, that force of blood pushing? Did that change? Yeah, that's the one that changes. So that's why we're picking a specific end, is because that force changes depending if we're talking about this end of the capillary versus that end of the capillary. So we're drawing our tissue. We have our interstitial space. We have the blood in the capillary, specifically the plasma. We have some fluid here, interstitial fluid. So we want to add on all of our pressures and think about them. So our pressure of the interstitial fluid. Okay. Which direction is that going? So our hydrostatic pressure from the interstitial fluid, where does that come from? Where does it go?
To the category.
Yeah, exactly. So we're thinking about fluid in this space. Fluid pushes on the walls of a container. So fluid in this space pushes in. Okay, we'll draw that arrow all the way there. Okay. So then we have our pressure, hydrostatic pressure from the capillary. Okay. So blood inside the capillary, what direction does that push? Okay, yeah, it pushes out. Okay. These are all happening at once. I'm just drawing them in order so that we can separate them out and see the arrows. Okay, so then we want to think about the oncotic pressure of the interstitial fluid. So pi F is due to solutes. So what direction is this arrow going? Out, perfect. Okay. Out, which means our final one, our pi cap, due to proteins in the plasma, okay, is going to pull in. Okay. So fundamentally, these are the directions of all our forces. When we calculate our net filtration pressure, which I'll write out now, net filtration pressure it's a pressure for filtration. So if it's positive, that means we have filtration. If it's negative, that means we have reabsorption. So what we're going to do is count up everything that's for filtration. So all our positive numbers for filtration. And then we're going to subtract out anything that goes the opposite direction. So we're still subtracting out all our absorption. So now all we have to do is substitute in these values in here. So we're just going to circle which goes with which. So filtration is that direction towards the tissue. Okay, so we're circling any arrows that go that direction. So we're going to get our hydrostatic pressure from the capillary and that oncotic pressure from the interstitial fluid. Those two arrows go the same way. All right. So our filtration part let's put it in parentheses just for neatness. So we're going to have our pressure, hydrostatic pressure from the capillary, plus any oncotic pressure from the interstitial fluid. And then we're going to subtract out anything going the opposite direction. Okay, so I'll pick blue. So we're just looking for arrows going into the capillary because that's our absorption direction. So we can see we have pressure from our interstitial fluid here, the hydrostatic pressure of essentially water fluid pushing back in. And we have that pull from solutes. Okay. So we're going to subtract out those properties. Okay. So we're going to subtract out the pressure of the interstitial fluid. We could add together in that same direction. Pi cap. But remember, when it's in parentheses, this means we'd need to distribute that negative. So this is essentially saying subtract the hydrostatic pressure of the interstitial fluid, then also subtract our oncotic pressure of the capillary. So then we have to do out the math. So this is going to equal our pressure, hydrostatic pressure in the capillary, plus, which was 38 from here, plus our other force for filtration. So 1 for that osmotic pressure of the interstitial fluid. And then we're subtracting out our hydrostatic pressure of the interstitial fluid. So subtract 1. And then we're subtracting out our our osmotic pressure of the capillary. So subtracting 25. Okay. And somebody want to do that math out for me? I'll do it myself. I like writing down math. I realize that's easy math. Okay, so we got about 13 here, right? Everybody else got?
Yes.
Okay, great. All right, so that would be our total net filtration pressure in this situation. Okay, so that's how we're going to do these calculations as we move forward. So next time we'll practice with the venous end.